Our next speaker is a terrific new member of the Autism Consortium. Dr. Jordan Smoller is Associate Vice Chair of the Department of Psychiatry and Director of Psychiatric Genetics at MGH. He's also a professor of psychiatry at Harvard Medical School and director of the Psychiatric and Neurodevelopmental Genetics Unit at the MGH Center for Human Genetics Research. The focus of Jordan's research has been the identification of genetic determinants of childhood and adult psychiatric disorders. He and his colleagues have also been studying, it's a big word, pharmacogenetic predictors of treatment response and how advances in genetics may impact clinical practice in psychiatry. Jordan is also PI of the Autism Consortium's research database of 500 families, which is kept curated and made available as a resource for our members for their individual research studies. Please welcome Dr. Jordan Smoller.
Thank you, Dee Dee, and thank you all. It's really a privilege and an honor to be talking to the group here today. And what I would like to do in just the next few minutes is give you a little bit of a sense of a theme that is emerging now in genetic research and neuropsychiatry. And the bottom line of that theme is that genes that seem to contribute to neuropsychiatric disorders don't necessarily track with the clinical diagnostic categories that we use, particularly those embodied in the DSM, which I'm sure everybody is familiar with. So I want to give you a bit of a sense of that. At first I have to find the pointer here. Okay. So here's what I would like to just touch on. One is, very briefly, some of the advances in neuropsychiatric genetics that have happened in the last couple of years, which have been extremely exciting, and many of you are undoubtedly familiar because autism research has been at the forefront of this. Then talk a little bit about the notion of a phenotypic spectrum. That is, that the diagnostic categories, the clinical categories that we have are really not ones that have sharp boundaries. That may not be a surprise to folks. But related to that is a third issue, and that is that we're finding that the genetic influences on these conditions also transcend the clinical and diagnostic boundaries, and that may have implications, for example, for why some conditions often co-occur at a very high frequency. And then we'll talk just very briefly about implications, how we might take next steps to further characterize this. So the history of neuropsychiatric genetics really began with family and twin studies that were trying to determine to what extent does a condition run in families, to what extent does genetic variation or do genetic differences contribute to risk in the population. And what you see in this table is a list of just some disorders and their familial relative risk. That means the increased risk to, say, first-degree relatives of somebody who may have the condition compared to the risk in the general population. So how much more likely are you to be affected, for example, if you have a first-degree relative than if we selected you at random from the population? And then the second column is the approximate heritability. That is, how much do individual differences in genetics contribute to the distribution or the risk of the disorder in the population at large. What you can see is that for all of the disorders that are listed here, the familial relative risk is greater than 1, meaning that these conditions do run in families, and that's something that we've known for some time. But also that the heritability is, which can range from 0, meaning genetic differences among people don't really contribute, to 100%, meaning that it's all attributable to genetic variation. That number ranges fairly broadly here, but it's substantial for a whole variety of neuropsychiatric conditions. So we knew that for some time, and there had been, just to really oversimplify and shorten a history, maybe 15 years when it became possible to begin to look at genetic variation actually directly. Lots of studies that were attempting to identify some of the specific genetic contributions that these prior studies suggested would be there, and without terribly much success in this area. And then as you know, lots of major advances happened in our understanding of the genome, beginning with the first draft sequence of the human genome, and then projects like the HapMap project and the 1,000 Genomes project, which cataloged genetic variation in populations around the world, and more recently the ENCODE project, which has looked at the function of genetic variation. And these kinds of breakthroughs really enabled advances in gene discovery that have just accelerated tremendously in the last few years. And so, for example, if you think about common psychiatric disorders, things like schizophrenia, bipolar disorder, anxiety, and so on, you might say that before 2008, just 5 years ago, if you asked people in the field, is there anything you really absolutely agree on as a genetic contributor to common psychiatric disorders, the answer would be probably not. And just in the last 5 years, that has changed dramatically. You can see this inflection point here. I think something here is a pointer. That wasn't it. But part of what happened was that we began to see much larger studies, the ability to look across the genome in a single bound, as it were. And so now just, these are estimates, but the big genetics meetings happened just a couple of weeks ago, and the number is something like 160 or so, where there are specific variants that have been with very high degrees of statistical evidence associated with a range of psychiatric disorders. So that's tremendously exciting. Now, we also know that there's another curve, really, that has happened in the background over a longer period of time, and that has to do with the fact that our diagnostic categories that we use clinically are a little bit of a moving target as well. So what you see here is a graph of the DSM, the Diagnostic and Statistical Manual in Psychiatry, and what you're looking at with these two lines is the number of this is the number of pages in the book. This is up to DSM-4, and the number of diagnostic labels in the book. So this has also been a bit of a moving target, but it also suggests that the boundaries between these disorders are, and how best to define them are really still a little bit unclear, and we are tweaking and trying to refine and lumping and splitting and so on. That may in part explain the common co-occurrence of disorders. That is, it's not clear exactly sometimes where one condition ends and another begins. In the case of the autism spectrum, there are a number of conditions that are seen at frequencies that are higher than, say, in the general population, so common comorbidities, including ADHD and others, as you probably know. And there is some evidence that some of this comorbidity is related perhaps to genetic overlap among conditions. So here's these are data from a Swedish twin study, a fairly large Swedish twin study, and what they're estimating here is the genetic and environmental overlap in conditions that may be related to autism spectrum. And so what you see here is the red part of these bars is the degree that genetic influences are in common with autism spectrum disorder in this sample. ADHD, fairly high, tic disorders, learning disorders, developmental coordination disorder, and so on. And this is something that we see throughout neuropsychiatric conditions. This is a study that I just showed because it's so large, was not looking at the autism spectrum, but this is from a Danish population-based registry in which basically everybody born after about 1980 was included for about a 15-year period, almost 1 million people. And what they did was they asked, what's the frequency of a psychiatric condition if, say, one parent had a serious affective or psychotic disorder, or both parents did, or one did and another parent had something else. And the numbers here are not particularly important to know, but so this is the parental affection status. That is, two parents, let's say, who had a psychotic illness, one did, and so on. These are conditions in their kids: schizophrenia, bipolar disorder, schizoaffective disorder, another kind of affective disorder. And every time you see an asterisk here, it means that it's an increased risk. And the bottom line of that is that for a very broad range of conditions, there seems to be something running in families, and it's not necessarily specific to the condition that is first diagnosed in the family. And in fact, twin studies allowed us to estimate what is called the genetic correlation. That is, how much do genetic influences overlap among different conditions. And this is just a summary from lots of different twin studies, and these estimates are, vary to some degree. But what you're looking at here is, for example, the overlap in bipolar disorder and schizophrenia, which was estimated to be about 60% genetic correlation. Autism and ADHD and learning disorder, OCD and Tourette syndrome, and so on. So that suggested that there was this degree of overlap and that these conditions were not necessarily, at least from a genetic standpoint, so distinct. And in fact, if you tried to kind of draw the DSM from a genetic epidemiology point of view, that is using these kinds of family and twin studies, you might see something that looked more like this than the nice, neat categories that you would see in the book itself. So in the past few years, as I was mentioning before, it's been possible to begin to look at the molecular basis, perhaps, of some of this overlap. And what are some of the specific genetic variants and specific genetic findings that seem to have these diverse effects, or at least contribute to them. And in the first series of insights here probably came from the work on copy number variants, which, as you know, are deletions or duplications, often of large chunks of DNA sequence. And in fact, rare copy number variants had been, were shown several years ago and increasingly now to contribute to some neurodevelopmental disorders, including autism spectrum disorders and schizophrenia and so on. And in fact, the striking thing about this that's emerged is that many, perhaps even most, seem to be expressed in different ways. That is, they can be risk factors for multiple disorders. Doesn't mean that an individual person has multiple disorders, but that they can be a contributor to autism spectrum disorder, to intellectual or developmental disability, schizophrenia, ADHD, epilepsy. So where you see this shading, you have a copy number variant that has been implicated, either as a deletion or a duplication. A similar situation began to be seen looking at more common genetic variation, not necessarily the rare copy number variations, but the more common single nucleotide polymorphism, single base changes in the DNA sequence that are just frequent polymorphisms or variations. And one of the first dramatic kind of demonstrations of the degree with which two categories which may have been thought to be very different seem to share some genetic basis came from a study published in Nature a few years ago from the International Schizophrenia Consortium. And this is a confusing slide, perhaps, if you haven't seen one of these kinds of analyses before. But basically what happened here is a genome-wide association study looking at markers across the genome, SNPs, was conducted in a large sample of patients with schizophrenia and unaffected controls. A score was developed based on that genome-wide association study. So taking the top few thousand most associated, even if they weren't strongly associated, variants, and essentially making a score out of that ranking, and then applying it to a totally independent sample. And the first question to ask is, well, if I generate a score of variants that may be associated, broadly speaking, with schizophrenia in one sample, do we see that same score accounting for some of the risk in an independent sample of patients with schizophrenia or controls? And that turned out to be the case. There was a strong, significant association between a genetic risk score in one sample, even though it wasn't predicting all of the risk in the second sample, it was strongly associated. But the sort of surprising thing, now not so surprising because lots of work has supported this, was that when a schizophrenia genetic risk score was applied to a bipolar disorder sample, there was also a strong statistically significant association, suggesting that there was some overlap in the polygenic, the many variants that were contributing to schizophrenia, some degree of overlap with bipolar disorder. And what you see in these last columns is the fact that for other non-psychiatric disorders like Crohn's disease or hypertension or rheumatoid arthritis, et cetera, you didn't see that. So it wasn't some general property of the fact that genetic risk scores simply are not that specific. So in the past year, it's been possible because of a tremendous sea change in the culture of how genetic research in many ways has evolved. And that sea change was a real determination to collaborate and to fuel discoveries by putting data together because of this realization that many of these effects, for example, are very small. And to be able to identify them, one needs to have extremely large study sizes. And so, as you may know, there was a consortium put together called it's called the Psychiatric GWAS Consortium, now called the Psychiatric Genomics Consortium. And initially, this was a consortium of investigators from many countries that put together data for 5 different neuropsychiatric conditions: autism, ADHD, schizophrenia, bipolar disorder, and depression. This sample has now increased well beyond the first wave. What this did was allow researchers to come together and discover new variants that wouldn't have been possible looking separately. It also created the opportunity to address the question that I've been talking about in some more detail, and that is, how much overlap might there be in the genetic basis of, for example, these disorders? And so the cross-disorder group of this consortium was able to look across these 5 disorders and basically ask a couple of questions. Oh, I'm using the wrong advancer here. Okay. So the first question we referred to as kind of a bottom-up question. If you essentially did the opposite of what psychiatry has done for the last 5 years, which is to kind of split categories more finely, and instead said, what if you combined these 5 different conditions into kind of a single category? What would you see rise to the top? Would you see some variants that maybe contribute to more than one? And the second one was to say, because there had been some recent findings for schizophrenia and bipolar disorder, if you looked at variants that had been associated with one of those disorders and now looked across a broader range of disorders, would you see association to this broader range? And I won't go into too much detail on the on some of the methods here, but I'll tell you that this report came out in just this last year, and it actually included a total of about more than 60,000 cases and controls. And what you're looking at here is a Manhattan plot, and for those who haven't necessarily looked at these before, this is just a representation of the findings of a genome scan in one picture, where across the bottom you have the chromosomes lined up, and each of these little dots, which sometimes coalesce into blobs, are individual genetic markers. And then on this axis here, you've got the strength of statistical evidence of the association, and this line is what's often considered significant after you've corrected for all the tests that you've done, genome-wide significant. And so the bottom line was that there were some findings that started to emerge in this bottom-up approach, that is, that seemed to be influencing more than one disorder. Here's just part of the rationale. So imagine that you had signals that didn't quite make it, perhaps, for one or more of these disorders, but that when you actually combine them, you would see something that rose to the top because there was this shared component and each of them was contributing. So there were 4, actually, individual variants in this analysis that seemed to have that pattern. And this is a busy slide, but I'll tell you that what you're looking at here is a summary of the results for each of these. So, for example, there was a region on chromosome 3, And what you're seeing here is for each disorder, ADHD, autism spectrum, bipolar, depression, schizophrenia, the effect estimate of association of this particular variant. And this is the total if you kind of put them all together. And so in this case, it appeared that this variant showed some evidence of association to all 5 disorders, whereas in this case, for example, the evidence seemed to be particularly marked for schizophrenia and bipolar disorder, but really not much for autism spectrum disorder, not so significantly for the other disorders. These were the top variants, and so, as you would predict, these are the ones for which most or all of the disorders were contributing. And I'll get back to a couple of interesting facts about these specific results in a moment, but you'll notice the similarity in the names of a couple of these, and that's because 2 of them were genes that encode calcium channels and are involved in calcium channel signaling, and this has become a theme as well that's emerged. So this was the other analysis, that is taking the top-down approach and saying, if we have variants that have been previously associated with schizophrenia or bipolar disorder in other studies from this PGC consortium, how do they seem to play out? And what you're seeing here is the best fitting model for what their genotypic to phenotypic effects might be. That is, if you look across the data, what are the data most consistent with in terms of the range of phenotypes or disorders that this variant might be associated with? So for example, for the 4 variants that were associated with bipolar disorder in previous studies, 3 of the 4 seem to be mainly specific to bipolar disorder. 1 of them seemed to have a broader, and that is this calcium channel, a broader spectrum. For schizophrenia, 7 of, it looks like 4 of them, I guess, were kind of specific, and the rest were actually broader. The other thing to notice in this slide and the last is this column, which is the odds ratio. That is the magnitude of effect. And these are relatively small. That is, this is a small increase, very consistent with what we've seen in other studies of the genetic, genome-wide association studies for other psychiatric and non-psychiatric conditions. The other thing that the consortium was able to do was something called a pathway analysis, which was to ask the question, if you, instead of looking at one variant or gene at a time, if you looked at genes grouped into biological systems or functions, were there any biological systems or sets of genes that are related that were overrepresented in some way in the results, that emerged as carrying more of the signal than you would expect by chance? And the answer was that there was one, and it was this calcium channel signaling pathway. So what that was saying is that there is evidence here that calcium channel signaling may be a pathway which has broad effects across these 5 disorders. And there's some further evidence now to support that as well. And then finally, looking at these polygene scores, remember I showed you this before with schizophrenia and bipolar disorder, we can now do it across the 5 disorders. So that is, take a genetic risk score developed from an analysis of schizophrenia. Is it statistically associated with the risk of, say, bipolar disorder or autism in another sample? And the bottom line here was that the greatest overlap seemed to be between schizophrenia and bipolar disorder. You can just tell that by the height of the bar here. But also there were lesser degrees of overlap between schizophrenia and depression, between schizophrenia and autism, between depression and bipolar disorder, and actually depression and, no, actually, I guess it's not in here. So, you know, mostly you saw it among these sort of more adult onset conditions like bipolar disorder, schizophrenia, and depression. That's in part because we were best powered to study those conditions because those were the largest contributing samples to this. So this was not definitive, but certainly provided interesting clues that the genetic influences overlap to some degree. And then in a subsequent analysis, this was led by Naomi Ray and Ken Kendler, also with the cross disorder group, using other methods that allow you to look at something similar to what a twin study can show you. That is, how much in an aggregate way is there a correlation between genetic variants contributing to one disorder and those contributing to another disorder? How much is overall their heritability shared? And the results here were quite interesting. So this is a table of the genetic correlation, similar to that figure I showed you before that was based on the twin studies with the little bubbles and lines between them. So for schizophrenia and bipolar disorder, it was quite high at 68, a correlation of 0.68. And that's high, but it's also consistent with what some of these earlier studies were suggesting. Lesser degrees between bipolar disorder and depression, schizophrenia and major depressive disorder, depression and ADHD, and smaller degree between schizophrenia and autism. Now, these are common variants measured. These are not copy number variations or rare variants. But what this sort of supports, now looking at the level of the genome, is that there are these shared genetic contributions. They're not complete, obviously, but they certainly do cross some of the diagnostic boundaries that we see. This raises a whole host of interesting questions to pursue as a next step. One of them that's interesting is, how does this actually work? How does it play out? Why is it that genetic variants might cross these diagnostic boundaries? One of the avenues that we're taking to study this involves brain imaging, and we've begun a reasonably large-scale effort to do this. And that's in part because there have been, this is probably a little bit hard to read, but what you're looking at here is some neuroimaging phenotypes or traits that have been studied, both structural neuroimaging and functional neuroimaging, most of which have been well established to be heritable themselves, and which have been associated with multiple disorders. And the question is in part the reason that we see genetic variation influencing multiple disorders mediated through brain circuitry that we might be able to observe that also broadly affects a range of neuropsychiatric phenotypes. And some of these things like intracranial volume and hippocampal volume now have large-scale genome-wide studies that themselves have identified variants that clearly contribute to those phenotypes. So at Mass General, we have been pursuing something called the Brain Genomics Superstruct Project with Randy Buckner and Josh Rothman and others. And this has made use of the fact that there are rapid acquisition protocols that can gather structural and functional brain imaging data fairly rapidly and on a fairly large scale. And this allows us to collect a lot of data relatively quickly. So in a few years, we were able to collect brain imaging, structural and functional data, DNA, web-based batteries on more than now about 4,000 individuals. And we have genome-wide common and rare variation data now we've just gotten and have not yet really analyzed for close to 3,000. So that's a fairly large sample. And one of the things that we're interested in is, can we use that to better understand how genes may cross these diagnostic boundaries? I'll show you an early paper that we did, which is on a much smaller sample than the one we have currently. And this was focused on the limbic system, the emotion regulation system, for lack of a better way of phrasing it. And in particular, the amygdala, a key node in this sort of emotional salience network, and the medial prefrontal cortex, which has been implicated in what's called negative affectivity or proneness to anxiety and depression. What we found was that in a fairly large sample, so it's about 1,000 folks who had had brain imaging, who did not have any kind of neuropsychiatric condition, a fairly robust association between amygdala volume, larger amygdala, and thinning of the cortex in the medial prefrontal cortex. And interestingly, that same area of thinning was associated with how well people could recognize emotions, particularly fear and neutral emotions, how well they classified them, and also a fairly broad range of measures of social functioning, including the broad autism questionnaire phenotype and measures of other measures of social functioning. And then we did this kind of analysis that I was just telling you about, this polygene score analysis, and asked, is there any association between genetic risk scores for any of these other disorders and this phenotype of prefrontal cortical thinning, and did find at least a statistically significant association that was actually, in this case, restricted to genetic risk scores derived for depression. Autism spectrum was not on this. We didn't have the data available for this. But we're now sort of taking this to, as I mentioned, a larger scale to be able to try to tease apart the relationships between genotype, psychological traits, and neurocognitive traits, and brain structure and function. So I'll wrap up by just summarizing that this is really a very exciting time to be in this field of genetics in neuropsychiatry. And we have really sort of seen the beginnings of a turning point, by far not close to the end of the story in understanding the genetic basis, but really something quite different from what we had 5 years ago. But this theme that's emerging, which is fascinating, is that the genetic risk variants, at least some of them, don't map onto clinical categories exactly, but are perhaps affecting broader systems. Calcium channel signaling in particular has emerged as one that may be involved in the pathogenesis or development of a range of neuropsychiatric disorders. And the hope is that as we begin to fill this picture in, as more and more variants come, as they coalesce into biological pathways, as we see what the architecture is of the genome and its relationship to clinical and behavioral phenotypes, we're going to get a better understanding, first of all, of disease biology, but also new avenues to pursue for treatment, perhaps. And also, interestingly, something maybe more sophisticated about how we think about the spectrum of psychiatric disorders and neuropsychiatric disorders, where we draw boundaries. Can we use genetic and other information to help us be more reliable and consistent about how we think about the boundaries among these various conditions? And you saw that escalating DSM curve. Can we kind of turn that a little bit and make that less unstable? So there are lots of next steps. I think I'm over my time, and I won't talk in any detail about them, but this has begun a whole program of research among groups around the world, really, that are interested in this kind of cross-disorder effects. We see it in other medical conditions as well, and it's I think, a fascinating theme to keep an eye on over the next few years. So I just want to mainly acknowledge the Psychiatric Genomics Consortium, which has really been just an absolutely terrific collaboration. And there are, you'll see Mark Daly's name here, who has been a driving force in both leading the Autism Spectrum Disorders Group and the Analytic Group, and of course is part of the Autism Consortium, and a number of other people who really led some of these analyses. And I think I will stop there and just thank you so much for letting me talk today.